As you've heard from the title of my work already, I'm very interested in cancer. Cancer belongs to the leading causes of death worldwide, and I guess everybody of you, and also me, we all know somebody who suffers or suffered from this very diverse disease, resulting in uncontrolled cell growth, which can affect every part of the body. According to the International Agency for Research on Cancer, this year, so 2018, 9.6 million people will die from cancer. This means that 1 out of 6 deaths is due to this disease, and the prognosis looks even worse. For 2035, 35 million deaths are estimated. And due to this very high number, there is a lot of effort put into the development of anti-cancer strategies. One major drawback of conventional anti-cancer therapy is that the intravenously administered drugs do not selectively accumulate in the tumor tissue. Actually, both cytotoxic drugs and also targeted therapeutics do accumulate at a lower level inside of the tumor compared to other non-targeted organs. And this, of course, leads to side effects and to the risk that the treatment has to be terminated prematurely. A major reason for this reduced transport is the highly dysfunctional vasculature due to the chronically inflamed microenvironments and the highly proangiogenic profile. So you can see that research in this area is very interesting. So when I had to decide where I want to do my PhD, I had several cancer research labs on my list. But as you may have realized already, I started my PhD in a platelet lab. So I learned a lot about platelets, their signaling mechanisms, their very important role in hemostasis, but also in thrombosis. However, over the recent years, the research focus in our field shifted towards the function of platelets beyond the classical hemostasis and thrombosis. So it was, for example, shown that they also play an important role in the innate and adaptive immune system, in the development of lymphatic vessels, or in angiogenesis in general. But I could never really give up my interest for cancer. And while we were studying platelets, we came across some very interesting reports that platelets also play an important role in primary tumors. So it was previously shown that when you deplete platelets in tumor-bearing mice, there is a huge amount of hemorrhage going on inside of these tumors. And it was also shown that when you deplete platelets, more chemotherapeutic agent accumulates in these tumors, which is very important due to the problems of chemotherapy that I explained to you just a few minutes ago. But you may appreciate that depleting platelets is a very good tool in experimental animals, but nobody would ever think of treating patients or depleting platelets in patients due to systemic bleeding complications that may occur. So we were interested in the mechanism, how platelets maintain vascular integrity inside of the tumor, in order to develop potentially later an agent that has, on one hand, an influence on the vascularization in the tumor, but on the other hand has no influence on hemostasis. And since my lab works for quite a long time with platelets, we already had a very good educated guess, I would say. And this was the glycoprotein 6. It's a protein on the platelet surface that has been shown to be important in maintaining vascular integrity during inflammation, for example. And it has also been thought of GP6 as a potentially safe antithrombotic target because its blockade has only very minor role in hemostasis. So we wanted to elucidate the role of GP6 in maintaining vascular integrity inside of the tumor. And you know, this sometimes feels like being a police officer trying to catch a serial killer. In our case, cancer would be the serial killer, but let's assume he's just too intelligent to prosecute it directly. So we have to use some tricks and tools to get a little closer towards our serial killer. And we suspect GP6 to be an important supporter of our serial killer cancer. Luckily, we do have very good tools to investigate the role of GP6 in our institute. So we do have GP6 deficient animals present, but we also have an antibody present that can functionally block GP6. This antibody is called JAK1, and when we use the Fab2 fragment of this antibody, this has no influence on platelet count in these animals. But there was obviously something missing in our institute, and that was the cancer expertise. So we looked around a little in the University of Würzburg, and luckily we found that we have very good cancer people in the Institute of Anatomy and Cell Biology in Würzburg. So we teamed up with Eric Henke, who was immediately very interested in the story after we've explained all this fancy platelet stuff to him. But you know, collaborations are not always easy. So the difficulty in this one was that both institutes are quite a few kilometers apart from each other, so I had to travel back and forth quite often, even sometimes 2 or 3 times a day. But you know, that keeps me active, and being active is said to be a very good cancer prevention, so probably my health will thank this collaboration in the future as well. But anyway, together we developed 2 different tumor models in which we wanted to study the role of GP6. One is the TRMC1 prostate cancer model, where we inject the cell lines subcutaneously at the back of the animals. And the other one is the 83 breast cancer model, where we inject the cells orthotopically into the mammary fat pad of the animals, so we can measure tumor growth. Due to the sake of time, I will today present to you the results of the 83 model, but I think it's very important to mention that we got comparable results for both of them. So as a start, we injected our cell line into wild-type and GP6 deficient animals and measured tumor growth. However, we did not see a difference. But after we terminated the experiment, we had a look at the tumor. And you may appreciate that the tumor in GP6 deficient animals look a little more bloody than the ones in the wild-type animals. So this was a first hint that GP6 probably is indeed a supporter of our serial killer. But to better resemble the clinical situation, we wanted to use our JAK1 antibody. So this time we injected wild-type animals with our tumor cell line and waited until the tumor is established. And then we separated them into 3 groups. One was the control group. The second one was a group which we treated with platelet depletion antibody. And you can see that when we inject the antibody, the platelet count immediately drops down to 0. And we could resemble the results present in literature already, that this leads to hemorrhage inside of the tumor. Now, when we use our JAK1 antibody, you can see that this has barely any influence on platelet count. However, we could also see increased hemorrhage inside of these tumors. So with this experiment, we could show that GP6 is a major player in maintaining vascular integrity inside of the tumor. But still, we don't know, does this now have an influence in the clinic or not? So again, we injected wild-type animals with our cell line and waited until the tumor was properly established and then treated the animals. But this time with a combination of GP6 blocking antibody and a chemotherapeutic agent that is widely used, and it's called Doxil. And after 2 hours, we looked inside of the tumor, and we saw indeed a slight increase in chemotherapeutic agent after GP6 blockage. But probably even more interesting is that when we look after 24 hours, in the control group, you can see that the amount of chemotherapeutic agent decreases clearly again. However, this is not the case when we block GP6. But still, we don't know anything about tumor growth now. So therefore, we used the same model, and as soon as the tumor developed, we started to treat the animals. These graphs show the treatment of the control, the platelet depletion, and of JAK1 FAB2, and you can see that only the single treatment with GP6 blocking antibody reduces our tumor growth. However, very interestingly is when we combine the treatment of GP6 blockade with chemotherapeutic agent, you can clearly see that we dramatically decrease tumor growth. So with this experiment, we show that when we block GP6, we can increase the efficacy of chemotherapy in these models we used. So to sum this up, we can say that GP6 is indeed a major regulator of maintaining vascular integrity inside of the tumor. So it is a supporter of our serial killer cancer. And when we block GP6, we can increase the efficacy of chemotherapy. So by locking away GP6, we can come one step closer towards our serial killer cancer. But it might take me a few more kilometers on my bike, traveling back and forth to elucidate the whole network of crime behind these murders. And with this, I'm at the end. And I would like to thank especially Bernhard Nieswand, Emina Mamadova-Bach, and Erik Henke for their great support over the last 3 years, and you for your attention. Thank you very much.